Hi, my name is Dorit, and today I'm going to present to you ReSynapse Therapeutics. We are developing multi-target drugs for psychiatric disorder enabled by computational drug discovery platform. The human brain is complex. The brain signaling system is composed of many proteins connecting with each other. In psychiatric disorder, multiple biological pathways are out of balance simultaneously. For over 25 years, PTSD solutions have been stuck because inefficient solution, and existing medicine are also insufficient. The core gap in PTSD is not lack of biological target proteins. The proteins are known. The therapeutic challenge is which protein combination to engage and how, based on the biological of trauma. What has been missing is a scientific framework to select the right combination and then to target them selectively. In clinical trials, because of this complexity, trial after trial has been failed. But in PTSD, the clinical outcomes, data has been accumulated, and protein function were revealed. By integrating the clinical and mechanistic data, decode which pathway create efficacy and which create side effects. We use this clinical outcome, we saw a map. What works, what fails, and how to avoid molecules that accidentally hit the wrong targets. By decoding this data, we have identified 4 proteins as an optimized combination. We have selected 4 proteins of serotonin targets based on core trauma biology of therapeutic benefits, while also intentionally bypassing the dopamine, adrenaline, muscarinic, and histaminic pathways that cause severe side effects. 2 of the targets are mechanistically involved in re-experiencing episodes in hyperarousal and post-trauma. So all in all, the biology defines the combination, and then what we need to do is to find chemistry that builds the single molecule to match this combination. This is what we did next. The first one was to define the 4 targets, which include the efficacy without the liabilities. Next, we moved to computational chemistry, multi-target drug design. The first thing was to identify a unique signature to all those 4 targets that differentiated and enabled selectivity. Next, we moved to computational chemistry in silico screening. We started with 40 million compounds, ending up with 250 candidates, from which 80 were synthesized for in vitro validation. In contrast to trial and error of traditional chemistry, out of the 80 compounds that we have designed, 12 have validated as multi-target compounds, and 1 is active on all 4 proteins. This is our data is experimentally verified externally, and our IP is provisionally patented in 2 patents. One of them is the combination and the computational methodologies, and the second one is a new chemical entity. I'm Dorit Cohen-Carmon. I'm a PhD by training. I'm a neurobiologist with over a decade in small molecule drug discovery and drug development. And with me is Itai Bloch, 15 years of experience as a computational chemist and computer-aided drug design. We are backed by a scientific and clinical advisor, Professor Alexandre Varnek as chemoinformatics, Anthony Alter, psychopharmacology, and Professor Yossi Zohar, psychiatrist, MD, PhD, the head of Israel National PTSD Center. We got NUFA IIA grant. PTSD is only our first indication, and we are expanding to treatment-resistant depression, which shares the same imbalance of serotonin pathways. This is a platform that enables additional psychiatric indications that require multi-target pharmacology. I want you to remember that each indication represents an independent asset, while the platform generates a pipeline. In order to cross our next milestone, we are fundraising $1.5 million for animal POC and lead optimization that will enable us to achieve the value inflection point for pharma towards licensing. We have no academic licensing agreement and no royalty obligation.
No need.
מה זה?
You don't need.
Yeah, we don't need. So join us for selective multi-target drug for neuropsychiatry. Thank you.